Okay, in this video I want to talk about protein sequencing, which I think goes hand in hand with protein purification. So I'm going to start out by just talking about a disulfide bond, because we haven't spoke about it, but it will become important in the sequencing technique. So a disulfide bond is a covalent bond formed by a coupling of two thiol groups. So essentially what that is we have two cysteines. So if you remember CYS, we have N CH, COO minus, and that's a CH2, SH is the amino acid cysteine. So if I were to draw like a disulfide bond, I would say, well, maybe here's my polypeptide, something like this, and then I would say, well, we'd have a CH2, an S, and another CH2. So essentially this bond formation here is a disulfide bond. And that's a covalent bond, so it's pretty strong. So don't get it confused with hydrogen bonds or any of the other weak intermolecular forces. We're dealing with a strong covalent bond. So it's important. Just understand what that is. So going on to the problem here, this is just the background information for the problem. And it says the peptide hormone glucagon from Nile tilapia was treated with chymotrypsin, and the resulting fragments were sequenced. A second sample of the polypeptide was treated with trypsin, and the fragments were sequenced. So essentially what we have here is two samples that were cleaved using one using chymotrypsin, the other using trypsin, and they produce these fragments. So the first part of the question, which I actually prepared an answer for because this is kind of more or less conceptual information here. It's kind of hard to write this whole thing out for you guys, so I'm just going to talk about it briefly here. And what they wanted you to do was outline a process by which a sequence of fragments of glucagon were determined from an intact polypeptide. So starting with the full intact polypeptide, the first step is to reduce any disulfide bonds by using something like 2-mercappa ethanol to separate the chains. So that's why I showed the disulfide bonds, because we're going to be breaking the bonds. We should at least know what they look like before we're breaking them. After the separation, after separating the chains, the amino acid content should be determined. Several samples of glucagon will be needed. Cleave one sample with chymotrypsin and purify the resulting fragments. So what's going on there? Basically what's going on here is we're setting up two separate samples. One of them we're going to cleave with chymotrypsin. Then we're going to purify the fragments. And to do that, each fragment can be separated using Edman degradation. The general process of Edman degradation is to react each purified fragment with phenyl isothiocyanate, forming a derivatized N-terminal amino acid. This can then be cleaved off, leaving the rest of the chain intact. So you react it with this phenyl isothiocyanate, and it basically becomes a derivatized amino acid, and it can be cleaved off, just one amino acid at a time from each fragment. So this can then be cleaved off, leaving the rest of the chain intact. Detection with phenyl thiohydration is more feasible than the normal amino acid, since it will absorb light unlike a typical amino acid. So once it's derivatized, it absorbs light. This is a way of finding the amino acid. And by running the derivatized amino acid through a column, typically high-performance liquid chromatography, it can be identified. And alternatively, mass spectrometry can be used to identify the amino acid, if you'd rather use that technique. The process is then repeated for the next amino acid in the chain to determine the second amino acid's identity. This continues until the entire fragment has been sequenced. So essentially, if you were asked to write out the entire process on an exam like I was, this is what you'd want to do, or something to this nature. Maybe not as detailed. You might just say something like, the derivatized amino acid absorbs light, which makes it easy to detect or something like that, instead of going through this whole bit about the actual technical names and everything for what's used, what you react the fragment with, this and all that. So I mean, this is just a general description, or how I would describe it. So the next part of the question asks that we then go back to this information and we sequence glucagon. So they want us to use what we have here, these fragments, and they want us to sequence them. So what I would do first, if I was doing this problem, is I would first look at both of these fragments, and I would look specifically at the first amino acid in all the fragments. So I would look at the first amino acid in all the fragments of trypsin, and I would say, is the first amino acid in any of these fragments the same as the first amino acid in any of the fragments cleaved by chymotrypsin? And if you look at that for a little while, you'll see that this histidine here and over here is the only amino residue that's first. Is the only amino acid residue that's in the first position in both the fragments. So that means we can kind of take that information now and say that that is the first amino acid in the polypeptide. And then from there, once you have that, you essentially just want to overlap the sequences. So the rest of the sequences will overlap. So what I do with these problems is I write down, say, trypsin, TRY for the trypsin, and let's say CHY for chymotrypsin. And once I have that written down, I will start with either of these two fragments. So for trypsin, I'll write the fragment in HE, SG, TF, SN, DY, SK. So I wrote that in, I'll cross it off now that I've used it, and what I'll do is I'll overlap it with the fragment in chymotrypsin here, which matches it. So it's HE, SG, TF. So now I got both of those done, and I look over here and I say, okay, well, SNDY. So I have that in my trypsin group, but I don't have it in chymotrypsin. So I look over here and I see, okay, SNDY. So I can cross that off and write that in here. Now I still have this SK on the end here, so I want to go back over here and look to see what I have from my chymotrypsin fragments. And what you'll notice is you have SKY, so I'll write that in. Now I have a Y in chymotrypsin, but I don't have it over here in trypsin. So I'll go over to my group over here and I'll look for the next one with a Y. And I'll notice that this has a Y. So I'll say, okay, well, that must be YLEDRK. And I'll cross that off. Now I'll go back to chymotrypsin again and I'll say, well, is there anything with this LEDRK? And there is right here. And I'll fill that in. So it'll be LEDRK AQDF. So then I can cross that off, and I've used that. Now I'll go back up to trypsin again, and I'll look over here. Well, what do I have? This AQ, D, and F, and that matches this one. So this is the overlapping fragment here, and that's AQ, D, F, V, R, W, L, M, N, K. So one of the things you'll notice about this right off the bat is that it takes a lot of space to write this fragment out, to do this overlapping sequence. So getting back to the problem here, we'll go back here and we'll say, okay, well, VRW, that takes care of this. Now we got the last sequence up here, which matches for chymotrypsin to this part of trypsin over here. So I will fill that in and that is L M N N K R S G A E. And then you can see over here RSGAA. So RSGAA. So that's it. By using the overlapping fragments, by using these overlapping sequences, essentially we were able to put the entire polypeptide together here. So after a fairly long and tedious process, you'll have the entire sequence for glucagon. So both of these are identical, they both match, and you would just say this is the sequence for glucagon. So that's it.